Hello guys, welcome to my YouTube channel. So today we are going to discuss RNA splicing. Now before going into the details of RNA splicing, first let us see the basics of that. So as you can see here, this is the genomic DNA, and from genomic DNA by means of transcription it forms the pre-mRNA. Now this pre-mRNA has two distinct regions as shown in this figure. This dark green regions are the exons. Alright, so these dark green regions are the exons, and just beside the exon you have a lighter green region which are the introns. So beside each exon you have one intron. So likewise this pattern is repeated throughout the pre-mRNA, that is one exon, then another intron, then one exon, then another intron, like that. So this sequence pattern is repeated. Alright, now as you can see on the right hand side also I have shown you another figure. So this is the pre-mRNA, and here the blue regions are the introns and the green regions are the exons. So what happens in RNA splicing, these blue regions or the introns are removed and all the exons are joined to form the mature mRNA. So this is the basic of RNA splicing, where all the introns are removed and exons are joined. Now these introns, why these introns are removed? Because these are the non-coding regions of mRNA, that is these regions do not code for a particular protein, and these exons are actually the coding regions of mRNA which actually code for a specific protein. So these non-coding regions are removed and all the coding regions are joined together to form the mature mRNA. Now let us see what is the structure at the intron-exon boundary. So as you have seen in the previous slide, this dark green region is one exon, and then the middle lighter green region is the intron. Alright, so this has been shown as the intron, and then you have another dark green region as the 3' end, which is another exon. So this is the notation that has been followed in this book. Now what you can see here is that at the center of the intron you have a nucleotide or adenine nucleotide. Now this position within the intron is called as the branch site, where this adenine nucleotide is present. Apart from that, what other characteristics you can observe is that this position is called the 5' intron exon boundary. Alright, so this is 5', I am writing it as 5' IEB, okay, 5' IEB, which is 5' intron exon boundary, and here also this position is called as the 3' intron exon boundary. So here I am writing it as 3' IEB. IEB stands for 3' intron exon boundary. Alright, now another thing that you can notice here is that near to the 3' end of the intron you have a polypyrimidine tract. Polypyrimidine tract means there are most of the nucleotides are pyrimidine. Alright, they contain pyrimidine residues at the 3' end of the intron. So this is the structure of the intron exon boundary. Why this structure is important, we will see that in the subsequent slides. Now let us see the basic mechanism of splicing reaction. Of course the splicing reaction involves a number of proteins, but first we will see the basic principle, how the splicing reaction happens. This is basically a schematic diagram without the proteins, and next diagram we will see with proteins. So what happens, as you can see, this is the 5' intron exon boundary. So this was your 5' intron exon boundary. Okay, so this was your 5' intron exon boundary, 5' IEB, and within the intron you have this A nucleotide or the adenine nucleotide at the branch point. So I am writing it as BP. Alright, so this is the branch point where you have the adenine nucleotide, and it contains a 2' OH group. Now here, as you can see, near to the 5' intron exon boundary you have a phosphate group attached to a guanine nucleotide. So what happens is that this 2' OH group of the A site attacks as a nucleophile to the phosphate. So this 2' OH group does nucleophilic attack to the phosphate. As a result, what happens? This phosphodiester bond that was present here previously, this phosphodiester bond is now broken, and a new phosphodiester bond will be formed between the 2' OH and the attacked phosphate. Okay, so what happens after this phosphodiester bond is broken? So this position of the exon is removed. This position of the exon is removed. So here, as you can see, this is the exon that has been removed, and the new phosphodiester bond has been formed between this 2' OH and the phosphate group. So here, as you can see, this phosphodiester bond has been formed within the intron between the 2' OH and the attacked phosphate. So this is the first step. Now what happens in the second step? As you can remember, this was your 3' intron exon boundary. Alright, so this was your 3' intron exon boundary. I am writing it as 3' IEB. So now what happens? The released portion of the exon of the first step, this contains an exposed 3' OH group. Now this OH group does a nucleophilic attack to a phosphate group near to the 3' end of the intron exon boundary, which is attached to a guanine residue. So this OH group does another nucleophilic attack to this phosphate group. So similar to the previous case, here this phosphodiester bond is now broken, and the new phosphodiester bond will be formed between the 3' OH of the exon and the attacked phosphate. So this phosphodiester bond is broken, and this phosphodiester bond will be formed between these two, this 3' OH and the attacked phosphate, the phosphate group that has been attacked. So what happens? This first portion of the exon, alright, so this first portion of the exon gets attached to this second portion of the exon by means of phosphodiester bond formation. So these two are now clubbed into a single spliced exon, and the intron is released as an intact lariat. Okay, so why this is called as lariat? Because it is sigma shaped. So in this way, the two exons which were previously far from each other are brought close together, and they are joined by means of phosphodiester bond formation, and the intron is released in the form of a lariat. Now let us see the individual proteins which actually bring about this splicing reaction. So first, as you can see in the first figure, there is a protein called as a U1 SNRNP, which is sitting at the junction of the 5' intron exon boundary. SNRNP stands for ribonucleotide proteins. Okay. Now, and apart from that, you have a BPP protein, which is branch point protein, which is sitting at the A site, and other subsidiary proteins. So in the first step, this BPP protein is released. Okay, and simultaneously another protein called as the U2 SNRNP comes and sits at the branch site where the BPP protein was earlier sitting. So this is the first step. In the second step, what happens? These subsidiary proteins are released, and a group of proteins which are U4, U5 and U6. So these three SNRNP proteins come together and they join with U1 and U2, which had been sitting earlier. So U1 and U2 were previously there. Now they are joined by the three proteins U4, U5 and U6. So here, as you can see, these three proteins have joined these two proteins. Simultaneously, you can notice that the introns are coming closer. Okay, that means lariat formation has just begun, and the next, after lariat formation, what will happen? The exons will be joined. So in the following step, what happens? This U1 protein is released from the complex, and as a result, now four proteins remain: U2, U4, U5 and U6. And also at the same time, the introns are now closer than the previous case. Okay, So they have come a bit more closer here. Okay, and then what happens in the following step, U4 protein is also released along with formation of this first portion of the exon. So the one thing has happened, first reaction of splicing has happened, the one part of the exon has been released. Okay, now in the subsequent step what will happen, this exon will act as a nucleophile to attack at this end, 3' end of the intron exon boundary. So what happens in the subsequent step, this released exon of the first step acts as a nucleophile to attack at the 3' intron exon boundary. So the first portion of the exon gets connected to the last portion of the exon. So the two exons are joined and the intron is released in the form of a lariat. So this is the spliced exon. Okay, so this is the spliced exon where the two exons have been joined together. This is the spliced exon where the two exons have been joined together by means of phosphodiester bond formation. So this entire mRNA that is released, finally this is all the regions are coding regions. Okay, and the intron or the non-coding region is released in the form of a lariat along with the three proteins which are U2, U5 and U6. So one thing you remember, that U2, U5 and U6, these two proteins were till the end of the splicing reaction. Sometimes question have been asked that which are the three proteins which remain till the end of the splicing reaction. So these three are the proteins which remain till the end of the splicing reaction. So this is the basic mechanism how splicing reaction happens.